So welcome to the first exam one review session. I decided to divide it up instead of trying to do everything in the 1 hour. I have a session in the afternoon, this session, and a session in the evening, and both will be recorded and they're not going to be the same. So in the first one, I present kind of an overall review of everything. And then in the second one, that's when I'll spend more time going into the more specific details, okay? So overall, exam one, and again, anytime during this session, if you have any questions, just type it into the chat or unmute and ask. It's easiest for me to see it if you unmute and ask, right? Because I don't have the chat screen up. Okay. So the exam one review session, your exam one is going to include the chapters on introduction, chemistry, cells, tissues, and the integumentary system. Okay, so these 5 chapters. Okay, we're going to go through the logistics. So for lecture, the lecture exam has 50 questions. And when you start the exam, the very first question is the same first question that you've seen in all the others, pretty much just stating that you know that you're not supposed to use any outside materials. You shouldn't have your phone, your book, or anything. It's just you and the exam. So that's the very first question. And then the next set of questions that you're going to see are all going to be multiple choice. So the first 40 questions that you see will be multiple choice. And the last 10 questions that you will see will be fill in the blank. Okay, a very valid question that I get is, what should I focus on? Okay, and essentially anything that we talk about in the lecture and lab videos or the materials that we give to you on Canvas, all of that is fair game. And we essentially draw questions from them and put them into a question pool. So I can't even tell you what question you'll get because each student gets a random pool of questions pulled from that. But what I can tell you is that we divided it up so that each section, so for example, you will for sure see 8 questions from the intro. You will for sure see 8 questions from chemistry, 8 questions from cells, 8 questions from tissues, and 8 questions from integumentary. So we divide it up so that all of the chapters are equally represented on the exam. Questions? Okay, so that means if we have 1, 2, 3, 4, 5 chapters, and I have 50 questions, so that means that there are about 10 questions per chapter. Okay, so I can't tell you what exactly to focus on because all the materials that we went through in the lecture and lab videos, all of that is fair game. Okay, and then we just kind of plop those questions into a pool and then it randomizes from each of those pools. Okay, all right, so for the lecture you'll have 50 questions, 40 multiple choice, 10 will be fill in the blank. That means 8 multiple choice questions on each chapter and 2 fill in the blank questions on each chapter. Okay, how much time do you have to take this?
Fifty minutes.
Yes, so it comes to 1 question per minute. And a lot of times that causes students anxiety, but I'll tell you this. So I did a study in terms of how much time students use. On average, students only use about 35 to 40 minutes on each of the exams. Okay, so just take it, you don't have to be like 1 minute, 1 question, 1 minute, 1 question. 50 minutes is on average more than students use. Okay, some students you take like 20 minutes, some students take 30 minutes, some take 40, some take the full 50. Okay, you should try to take the full 50 because that means that you're processing the question with adequate time instead of like speeding through it, because the more you speed through it, the more likely you are to make mistakes. Okay, so you do have 50 minutes, but I will let you know that on average, students take about 35 to 40 minutes, even though I stress to them, slow yourself down, read the questions out loud, make sure you use the full 50 minutes. Okay, so 50 minutes on the lecture exam. Okay, you will see one question at a time. Okay, you can move back and forth, but I do not recommend that. And the reason why I don't recommend that is because it's just too stressful to click back, click forth, and then try to remember which question you answered, which one you didn't. Even though as you're taking the exam, it will show you which questions you complete and which ones you haven't. Okay, it's just easiest to spend the adequate amount of time on each question and keep moving forward. There's a benefit to that, because if something should happen and you get kicked out, I will go in and see if you've already seen a question, I can't let you retake it because you've already seen it, right? And I can't have the question be changed, okay? So if you've seen the exam, I can't. So if something happens and it's a legit reason, but if you've seen the whole exam, I can't let you back in because you've already seen the exam. So it's best just to take it one at a time and then spend the adequate amount of time on them. And then if something should happen along the way, technical difficulties, and you get kicked out, I'll go, oh, okay. Susan only got to question number 17. She only took 20 minutes. She still has time left, and she hasn't seen any of the other questions. I'll try to see if I can open it up to question 18. And then you can take it onward. Okay? So that's why it's easier for you just to take one question at a time, answer it, and then move on to the next one instead of skipping around. Okay. Questions? What should you do if you do encounter technical difficulties?
Text or call?
Yes. Please text me. Don't email me, because I won't, I won't always be checking my emails. Just text me. Okay. So if something happens while you're taking the exam, text me as soon as possible. Okay. So text me as soon as possible and let me know. Okay, so that's a good time to text me is when you're taking your exam. Okay. Other things I prefer email because it's not urgent, but when you're taking the exam, that could be urgent because you have a smaller window of time to take the exam. Okay. So if something should happen, just go ahead and log out and then text me. Okay. What else? Any questions you may have? Okay. For your lab exam, okay, lab exam is 30 questions. Okay. And it's going to start from all the intro stuff. That's a lot of vocab. If you remember that lab, the first lab is a lot of vocab. So you can expect that to be on your lab exam, as well as the cell images and the tissue images. So these three things are going to be what will be on your lab. So the histology, the slides for the tissue, the slides for the cells, and then all of those terminology, like anterior, posterior, pedal, axillary, so on. All of those terms will be on your exam, okay? Your lab exam. So the 30 questions on your lab exam are just like your quizzes. All of them are fill in the blank. You have to type in your answers, and spelling counts just like they do on your quiz. Okay. You will have 45 minutes to take this because it is all, you know, you have to type in all your answers. What else? Questions on your lab exam or your lecture exam? Okay. For both exams, okay, you need to have respondents locked down and the webcam is required with an environmental scan. Okay. So in the announcements page, I have an announcements for preparing for your first set of exams. There should be a video demonstrating what I'm expecting of an environmental scan. Okay, now I'm going to show you one of what is not acceptable as well that students have done in the past. Now, your environmental scan, I just wanted to let you know that the environmental scan, I never look at those. The only time I look at them is when or if the respondents lockdown flags you, and they flag students if the students are not in the camera or if they're looking around or, you know, there's an algorithm that helps them determine whether there is a discrepancy in academic integrity or not. Okay, so I only watch these environmental scans if you are flagged. Okay, so I don't watch them at all. Okay, but when you are flagged, the environmental scans are what will protect you. So if you're flagged and I look at the environmental scan and it shows that there was nothing around you, you know, I just go ahead and grade the exam as regular. Okay. So so the environmental scan really is meant to be your protective, what kind of keeps you, you know, free of, you know, potential academic disintegrity. Okay. So I'm going to show you what one would look like. Okay. So some examples would be students would go something like this. Okay, that doesn't tell me anything. So you'll find that in the environmental scan, what they'll say is that you can either have a mirror, a long mirror in the back to show what is, you know, what you're doing in terms of what's around your table and what you're currently doing. Okay. So other times students do the environmental scan like this, up and down. Okay, that doesn't tell me anything either. So what you'll want to do, or what I want to know is I want to know what's at your table. You know, oh, my drive connected, so it's a little bit more difficult. So what you can do is you can do kind of a, if you're, especially if you're on a swivel chair, you can do a 360 to kind of let me see what's on both your right and left table. You can see, and it should be clear. There shouldn't be anything around you. Okay. And then in the past, I've had students have their phone on their lap, and respondents will be able to track that because you'll be looking down in a weird angle. So just, you know, make sure that it's kind of like, oh, there's my drive. Um, just to make sure that there's nothing, show me that there's nothing around you. Okay. Questions? There's a video that demonstrates it much better than than I did, but I'm just showing you what students have done in the past that were not considered acceptable environmental scans. Okay. Questions? All right. Where's my this? Let me put this back up. There we go. All right. Any questions about the logistics? What's kind of being expected? what the exam would look like. All right. So kind of going through the same thing, but here you'll have it in writing. So the content lecture exam is going to be on intro, chem, cells, tissues, and intake. The lab exam will cover the intro, the terms, the slides of the cells and the slides of the tissues. Time frame, you have 1 minute to do your lecture exam, but this one we changed. Now you have 45 minutes. So 45 minutes to do this one. Logistics, make sure the respondent's lockdown is on as well as the webcam. Exams will not be graded unless you complete an environmental scan. And during your exam, your face must be fully on the screen. Respondent's lockdown will flag you or notify me if something if that doesn't happen. You will just unfortunately because you would have seen the exam, you would not be able to retake the exam and you would just be given a 0. In addition to that, I have to notify the academic dean, okay? And it could potentially go on your record. So some troubleshooting if something before you take the exam, practice using the practice quiz. That one you can take as many times as you want. It's more of just to make sure that your computer or whatever you're working on is compatible with Respondus Lockdown. So use the practice quiz before you take the exam to make sure that, for example, your internet is stable, Respondus Lockdown is working, and to test out the webcam function. Technical difficulties, don't wait until the last minute to take the exam. So sometimes students wait until Sunday at 10 p.m. to take the exam and something happens. And once you are beyond that time range, even if some technical issues happen, I can't help you, okay? So it's best to try to take it within that time a little bit earlier than when the absolute deadline is. Stay calm if something does happen and text me, okay? Then in terms of recommendations, in the announcements page, there should be links to what's called a 5-day study plan. Don't wait until the last few days to cram everything in. It does not work for this class. And the reason why is because there's just so much content in this class that it's best to spread it out. And it helps unburden the load of how much content, it's content heavy when you kind of categorize each chapter into its own day. So for example, you can study the oldest material first because that's most likely what it's further away, so it's less likely for you to remember it. Or you can study the hardest one first and then, move forward, but try to set yourself specific chunks to study each day. And that's why one of the very first assignments that you did for this class was time management. Okay, the time management students day is one of the best strategies to do well in the exam, is to actually put it on a calendar when you're studying what, how you're going to study it, and it keeps it it helps you to stay accountable. Okay, and so that's the 5-day study plan. Then studying, making sure you assess and review, and then make sure you cover the test taking strategies. Okay. Studying and reviewing are not the same things. Students think they are, but it's not. Studying is when you're putting the information into your brain to establish that network. But reviewing is practicing pulling that information out. So to review, I recommend a lot of you shared at the beginning that you are planning to do flashcards, Quizlets, reviewing your quizzes, your old quizzes. Quick question, how many of you have been able to pull up your old quizzes?
There's like a certain date point where you can pull them up, but then after like X amount of days, they're locked back out again.
That's weird. It should be set so that you can see them all the time. You just can't see the correct answers. So let me go into that really quick. Let's do assignments. Let's say, for example, your quiz 1. So it's set so that you can always see your review responses, but the correct answer should only be shown for those 2 days after I release your grades. So are you not able to see the correct answers, or are you not able to see the quizzes at all?
That's what I meant, that you could see the answers for only a certain amount of days. Sorry, that was
Oh,
confusing.
got it. Yep. So that's correct. So you should be able to see your quizzes though, and which one you got wrong. It just won't show you which one is the correct answer after those 2 days. Okay. And that's good. Okay. So make sure you go into your quizzes, see which ones you got wrong, and look at your notes and see if you can figure out what the right answer is. And if you can't figure it out and you're just like puzzled over it, send me an email. Okay, and I'll help you work through that content. Okay. All right. So all of you should be able to pull up your quizzes and see what questions you got, what questions you answered. You just won't be able to see the correct answers for them. Okay. All right. So use your previous quizzes as a measurement of, what you got wrong, which one you got right. Okay. There's two ways in which you can study the content. Either study the oldest material first or study the hardest material first. And studying complete chapters. Don't skip around because it gets a little bit overwhelming if you do that. Okay. Does anyone have any strategies that they found worked really well for them as you're preparing for the exam? Flashcards. Thank you, Anna. Yep. So flashcards, especially for the lab terminology, I would highly recommend doing that. Okay.
I'm
That is
also a
a,
flashcard. Oops, sorry, go ahead.
oh, no, I was just going to read Matthew's comment. Go ahead. What were you going to say?
I'm also a flashcard person, but I really like that Quizlet lets you play like games with the flashcards just to kind of give, kind of a fun incentive for winning those, learning those terms and winning the rounds and stuff.
Exactly. Plus, Quizlet is really nice because the ones that you answered right, it won't give you that again until closer to the end. It actually focuses on the ones that you got wrong and keeps on cycling those through. So it helps you really kind of figure out which ones you got wrong and help you do that more. How do you access Quizlet? It's a free app that you can download onto your computer, into your phone, but it's an app where there are previous questions I've already written, but I suggest you write up your own questions. And then it's like doing flashcards, but a virtual version of it. Okay. Yeah. All right. Thank you. And if you have any other strategies, share them with me, and then I'll send out a mass email to the class. Okay. So now we're gonna get started. And we're going to look at what kind of things you should know for each chapter. And again, this is a very broad overview. Okay, and like I said earlier, all of your test questions for both lecture and lab come directly from the lab and lecture videos, as well as the materials that we post for that. Okay, so that's your main source. Okay, now there are reading textbook pages. I would not focus on those first, except for lab. Okay, so really for lecture, you should 100% of the questions come directly from those lecture scripts, lecture videos. So that helps you narrow down a little bit at least. Okay. Because if you've had a chance to look at the textbook, it's a lot of information. Okay. All right. So for introduction, make sure you know the scientific method, all the terms and definitions. Here's a nice place to use flashcards, Quizlets. Okay. So know what all the terms for the scientific method are. Be able to define the difference between anatomy and physiology. Okay, so let's just practice. What's anatomy? Your body. Okay, good. but let's be a little bit more, try to be more precise in the definition. Because technically both of them have to do something with your body.
The various structures of your body.
That's right. So anatomy focuses on the structure of the body, where physiology focuses on the function, how things work. Okay, so if let's say you were studying the muscles of the hand, what would that be? What would that be an example of?
Anatomy.
Perfect. If you were studying the mechanisms of how your muscles contract, in order for your hand to move, what would that be? Physiology. Very good. Okay. All right. So you can see that your exam questions could be a straight regurgitation of what the definition is, or I can ask you to apply the question. So I can say, okay, muscles, if you were studying muscles of the hand, what would that be an example of? Instead of saying structure of the body means what? Okay. So there's several layers of which I can ask you these questions. One, the most basic is just regurgitation. Do you know the definition? And then the higher type of questions, the higher levels that I can ask you is more along the lines of, can you apply that definition? Okay. All right. So I have some in the chat. Perfect. Thank you, Anna. Thank you, Caroline. Okay. Then make sure you know the levels of a hierarchy. I actually had an email about this one that the students didn't see this in your notes. It's in there. So make sure you learn about what is the smallest to largest, or it could be largest to smallest. Okay. So we can go small starts from atoms to molecules to organelle to cell, tissues, organ systems, and organisms. So a test question might be, okay, I have these terms and I want you to put them in order. So you need to know that atoms is the smallest, then molecules, then organelles, cells, tissues, organ system, organism. Or I can ask you what's the largest to the smallest. So what was my key point in sharing that with you? What do you have to do to make sure you get the correct answer? You have to read the question. Okay. That I will share with you that like 45% of the time when students answer a question wrong that they knew, it's because they didn't read the question carefully. Okay. So make sure you read the question and make sure you know what exactly the question is looking for. The question could be list the following in the smallest to largest, or it could say list the following in the largest. Did I reverse it? From the smallest to the largest, or the question go from the largest to the small. So you really have to make sure you read the question. Okay. Yeah, and you also need to know the order. Anna's right. You need to know the order, but you also have to make sure you read the question. Okay. So this would be a level 1 type of question. I list the following and I'll ask you to put in the correct order. A higher level of the same question could be, okay, I've got the heart, I've got water, I've got epithelium, and I would ask you to put these in the order of smallest to largest. So from smallest to largest, how would you put this? What's the smallest? Water, very good, because that's a molecule. Good. What's next? Epithelium, that's right, because it's tissues. And then last, you'd have the heart because that would be an organ. Okay. So here, I'm not using those terms. I'm using examples that you would need to show that you understand what they are and how to put them in the order. Okay, so if you see an option where it goes water first, epithelium second, and heart third, that is the correct answer if I'm asking from smallest to largest. Okay, just kind of anticipate what kind of questions you'd get. All right. Okay. Then we have, oh, I need to move the chat away so I can write on my screen. The next one is homeostasis. You know you're going to encounter at least one question on this because homeostasis is a huge concept. If you had to leave anatomy, physiology, this class with just one main concept, it would be homeostasis. We will be talking about homeostasis the entire time. Every single chapter has something about homeostasis on it. It's so important. Okay, so make sure you understand the basics of homeostasis, the definition, be able to maintain stable internal environments. Okay, and then of course, that's just the very basics, right? You need to know how that happens. So what's the difference between negative feedback? What's the difference between positive feedback? The dynamic equilibrium. So there were a lot of terms that were discussed in here in order to get the full picture of what homeostasis is. Okay, it's a big concept that runs through every single chapter in this class. So make sure you know that really well. Okay, so dynamic equilibrium were some, set point was another term. Okay, so lots of terms that you need to memorize for chapter 1. Okay, now again, I'm giving you a brief overview. Okay, so anything in your on Canvas under the lecture video, lecture script is fair game. So use that as your studying guide. Okay. Questions?
I personally have a hard time with negative and positive feedback. Do you have any tips on differentiating those? Just, like little, I don't know, sayings or anything to help remember which is which?
It's not so much as sayings, but I think of it like if it's negative, it's going backwards. If it's positive, it's going forward. So positive feedback is amplifying. It's getting amplifying, amplifying. It's getting greater and greater. Where negative feedback goes opposite. So when it senses a change, it does the opposite of it. So I just kind of use the term itself to kind of help understand what negative and positive feedback. And you know what? I think that after I give you the general review, the rest of the time, I'm going to focus on that positive and negative feedback. So thanks for sharing that. I will make sure I cover that before we end today. Okay. All right. Then for chemistry, I'm personally weak at chemistry, so I would set aside more time to study this one. Okay. If you're good at chemistry, ignore this suggestion. I'm good at math, I'm good at physics, I'm good at biology. I am not good at chemistry. So it takes me a little bit longer to understand chemistry. Okay. That may be true for some of you, may not be true for all of you. Okay. But for here, make sure you understand the terms. What's an atom? What's an ion? And then within ions, there are two forms of ions. There's cations and anions. Okay. And a good way to remember that is cation. You see it has a plus right here in the T. So cations are positively charged ions, where anions are negatively charged ions. Okay. Make sure you go through molecules, what's a solute, and all the other terms that you find in chemistry, which is a lot. So again, this is where flashcards would be very useful. Okay. Then within molecules, you're going to want to be able to know what's stable versus what's neutral and what does it prefer. So let's just go through that, do that together. What does it mean for a molecule to be stable versus what does it mean for a molecule to be neutral? I can get us started. So neutral refers to, there you go. Thank you, Anna. So stable refers to when it has the full electrons. Full valence, let's be specific, valence electrons. And that's why knowing the terms is so important. You know, if you haven't got a chance to study this, you're like, what the heck is a valence electron? So it turns out that an atom has the center, and the center is the nucleus. This is not the same as the nucleus of a cell, mind you. This is the nucleus of an atom. So there's no DNA in here, okay? The only thing that's in here is protons and neutrons. Okay, so the nucleus of an atom contains protons and neutrons, and then surrounding it, they have these shells or orbitals that contain electrons. The first shell can hold up to 2 electrons. The second shell can hold up to 8. And then the third shell can hold up to 8 as well. So whatever shell its outermost shell, we call that a valence electron, valence electrons. The outermost shell contains the valence electrons. Okay, so if it has all of the valence electrons it can hold, in other words, if it's a third shell, how many valence electrons would make it stable? If it's the third orbital, third shell, how many electrons would make that stable? Perfect, 8. Okay, because the max it can hold is 8 on the third. Okay, if we're looking at the first shell, your answer would change to 2. Okay, thank you. So that's what it means to be stable. It has all the valence electrons it can hold. Where neutral means that what? Since I know you know it. What does neutral mean? Yes. Okay. So neutral means that the number of protons is equal to the number of electrons. Okay. How do you know how much protons it has? You do not have to memorize it. If I ask you a question, I will always give you the number of protons or whatever it is in the formula for you to figure it out. Okay, so you don't have to memorize the periodic table. Okay, so you don't have to memorize, okay, oxygen has this atomic mass, has this number of proton. I will provide that for you on the exam. Okay, but you do need to know, what does it mean to be neutral? So that means that the number of protons equals to the number of electrons. Okay, what's a chemical bond? And there's more, but I only want you to know the very basics, which include ionic bonds, covalent bonds, and hydrogen bonds. I can ask you for the pure definition of it, or I can give you some application. For example, which of the following is the strongest bond, chemical bond? What would you say? What's the strongest chemical bond amongst these 3? Strongest chemical bond, perfect. Covalent is the strongest, okay? Or I can ask, which of these bonds occur due to the transfer of electrons? Which of these bonds is due to the transfer of electrons? What would you say? Ionic, very good, okay? Perfect. So that's the type of questions you're going to see in the exam, okay? You're also going to see questions based on pH scale, okay? So pH scale is the measurement of acidity. How can you remember that? Is because it's the P, the measurement of H. What's H? Hydrogen. Okay, that's why it's the pH scale. It's a measurement of H, and H is the symbol for hydrogen. The more hydrogen you have, the more acidic something is. Okay, so the more hydrogen you have, the more acidic it is. The more hydroxide you have, OH, the more basic it is. A pH scale range is what to what? A pH scale range is what to what? Thank you. 0 to 14, which makes 7 neutral. 0 is acidic, and 14 is basic. At this point, you're like, wait a minute. Didn't you just say the more hydrogen ions there are? So in other words, shouldn't that number 14 be acidic? So why is it basic? Well, it's a mathematical thing where P is taking the logarithmic value of something. When you're taking the log of something, you're actually taking the reverse of it. So the greater the amount, the smaller the value. The greater the amount, the smaller the value. So actually, the closer to 0 it gets, the more hydrogen ions that are present. And that's why the more hydrogens there are, the closer to 0 it gets. In other words, the more acidic it is. Okay? Then make sure you know the organic compounds. Okay? And that's the 4 main basic ones. You tell me, what are the 4 main basic organic compounds? Come on, 4 main basic organic compounds. Thank you. Carbohydrates. And they gave us all 4. Carbohydrates, lipids, I gotcha. Proteins, and not nucleotides, but nucleic acids. Okay, if you remember, you had to do that cognitive map. That's so useful. And the reason for that is because rather than memorizing paragraphs by paragraphs about all of these, that cognitive map puts the information into boxes, and it's easier for us to study as well as pull out during the exam from our memory when it's categorized like this. So you can imagine you're taking the test, you're like, okay, I remember the carbohydrate box, and in that, here were the types. Okay, so the more cognitive maps you use, the lighter the load is on your brain, and your ability to pull the information back out becomes greater. Okay, and that's why, yeah, exactly. The maps really do work. And I'm not basing it on experience, I'm basing it on research, I'm basing it on experience as well, and just scores overall of students who do cognitive maps and students who don't. Okay? All right. So here's an example of one. Okay. So I've completely filled out the first one for you, carbohydrates. I left the last 3 for you to fill in yourself. Okay. You should actually try to do this from scratch. Don't use your notes. Just put down, just drop what you know. And then pull out your notes and go, oh, I only remember this, but I'm missing this. And that's when you fill it in and you make it a full cognitive map. Because 1, that is the process of studying. 2, that's also the process of reviewing, because now you're going to know what you knew from what you didn't know. And what you didn't know is what you should focus on. Okay, so you can see under carbohydrates, here are the functions: energy source, energy storage, and it actually is part of the cell structures as well. There are 3 types. There's monosaccharides, which has 1 molecule of sugar, disaccharides, which has 2 molecules of sugar, and polysaccharides, which is a long chain of glucose. There's glucose, fructose, and galactose as monosaccharides. So on a test, you could see something like, okay, I've got sucrose, I've got starch, I've got lactose, I've got glycogen, and I've got galactose. Which of the following is a monosaccharide? That's the type of question you'd see here. Or I can ask, okay, which of the following carbohydrate is ingestible? In other words, you can eat it and you can digest it. That would be starch. That would be another type of question I could ask based on this content. Okay, all right. So again, the purpose of this is just to provide you a general overview. I know that I talked about doing the homeostasis, but I don't think I'm gonna have time because I want to go through some, make sure I go through all of the overview first. So the second session this evening, if you can attend it, we'll actually go back and go into more details. Okay, so if you can't come to the second session, then I will record both sessions. Okay, but I just want to make sure I cover all the things you need to know for this first. All right, so for cells, make sure you know the structure and function. There was a worksheet that you didn't have to do, but you could do to study. The worksheets are really, I mean, a lot of students complain that they're hard and long, but guess what? They are one of the best studying tools you could have, okay? Especially if you do it in the way I'm going to tell you. So when you do the worksheets, do it twice. The first time that you do the worksheet, just use what's currently in your brain. Okay? Then the second time, pull out your notes. And you do this in a different color. That's going to allow you to know what you currently know from what you don't know. And you need to study this part right here that was in your notes, but not on the worksheet. Okay? So same worksheet. The first one, you just jot down what you currently know. And then the second time you do it, pull out your notes and then fill it out. And that's going to really help you assess what you know from what you don't know. Okay? Then make sure you understand the plasma membrane. There's a whole section on plasma membrane in terms of all the components and function. This is another hard concept that will, if requested, and I think you should because it's a hard one, for this evening is the mechanisms of transport. What's the difference between passive versus active? And then within passive, there's diffusion, facilitate diffusion, osmosis, and filtration. How are they different? How are they similar? Under active, there's vascular transport, which include endo and exocytosis, as well as just plain active transport. And then the organelles, all of you did really nice videos on that. So watch each other's videos, okay? And I know I owe some of you your videos, and any video assignments is going to take a little bit longer for me to grade because I have to watch them like 2 to 3 times each, and there's like 50 of you. So that's why it's taking a little bit longer to get back, but I will always get back your quizzes as soon as possible, okay? So watch each other's videos on the organelles because y'all did a really good job on that, okay? All right, then we have tissues. It seems to me out of all the chapters, students really struggled on the last two, tissues and integumentary. The quiz grades kind of dipped a little bit for these two. It's really not that hard. When I was first studying this stuff, it felt so hard because there's just so many. But once I put them into cognitive maps, it was much easier. So really, I highly recommend it. If you don't do any cognitive maps, except for two, these would be the 2 chapters I'd do them for. Okay, because this one is not heavy on the physiology as much as it is on the anatomy of it. Okay, so make sure you understand the 4 types. Oh, sorry. Let's start with make sure you understand the structure and function of the tissues, and then understand that there's 4 types: epithelium, connective, muscular, and nervous. Know how they're different and how are they similar. Okay, now of these 4, I went through the last 2 very lightly because there's another chapter, separate chapter on nervous and muscles, and that's why I didn't go into them in too much detail. What I did go into in a lot of detail, a lot of detail, is epithelium and connective. So you really want to focus on these 2. Okay. Then, so focus on these two. Now, some useful resources that you should see, especially for lab. Okay, I put up some supplemental videos on the announcements page, particularly for the lab, because a lot of you were concerned that there's some images on the quiz that you didn't necessarily see on the lab PDF. Go to my video. So the supplemental lab videos has all of them. So I highly recommend you using those. And they're posted under the announcements page. Okay, so let me show you what they look like in case you hadn't had a chance to go there. Let's go to announcements. Let's go to week 3. So I'm talking about these right here. The supplemental histology lab video right here. Because it actually walks through, and I of course can only share my experience, but how I was able to learn this material. Like, you know, I look for the basement membrane and I look for the free surface, and from there I count down. So I share with you my strategy in ways in which I learn how to identify these slides. So take a look at that. And I also went through, I also share a video of how I did the cell worksheet. Okay. So lots of videos, lots of resources. They're supplemental, but they're extremely useful because I made these specifically for this class for you know, this content, and they're not generic. Okay. So if you have time, please do take a look at those. All right. Then the integumentary system, same thing. This is probably one of the harder ones. I would, you know, if your strategy is to do the hardest first, I would do this first. It's just because it has a lot of content. Okay. Structures and function. Integumentary system includes the skin, nails, and hair. Don't forget these last two. Okay. Even though we do focus on skin. So in terms of skin, make sure you know the functions. It's the barrier. Make sure you know what an acid mantle is. Make sure you know what sweat is. What does it secrete? Okay. Then structure, the anatomy of it, know the layers. It's divided into cutaneous membrane, and technically that's what skin is. It's the cutaneous membrane that only involves the epidermis and the dermis, where the subcutaneous membrane, that's where your hypodermis is. And technically that is not skin, that's below skin. Okay. Then we talked about the epidermis, the different layers, stratum bacilli, spinosum, granulosum, lucidium, and then corneum. And again, if I'm asking you these layers on a test, make sure you understand if I'm asking it from deep to superficial or from superficial to deep. So read the questions carefully. Mechanto mnemonic, like one of my students, I can't remember her mnemonic, something about like bears, spit, grapes, something, but make a mnemonic out of it so it helps you kind of remember. Okay. I only know like the raunchy ones. Like when we get to the bones, the carpal bones, there's a medical school mnemonic where it says, some lovers try positions that they can't handle. So it like gives you the bones in the order and gives you the first letter of each bone. Okay. All right. So this one would be a nice cognitive map for. Okay. Like you did on the worksheet. Some of you chose to do this worksheet, and that's a good one to go to. Know the dermis, what tissues form the dermis, what are the accessory structures and the glands, and so on. And then don't forget the last one, which is membranes and glands. Okay. Yes, I agree. I love cognitive maps because it takes time to do, but when you're taking the test, it's going to be so easy to remember everything because you can just visualize what was in what box. Okay. And then we're going to end with this, how to prepare for lab, use the PDF, listen to the video, write the correct answer in your PDF, then use the PDF as a quiz. Okay. What to study for lecture, the PowerPoints, and the videos really are the main ones. So in terms of studying, rewrite your notes, do the cognitive maps, anything that's active and not just reading alone. If all you do is read your notes, you're not going to remember everything. It's just too much. And then practice recalling the information, do self-tests, and then make sure that you watch the video because there was a video that was created for how to take a test specifically in this class. Okay, so those are the very basic stuff that I'm going to share with you in this first half. In the second half, I'll go back and I'll go through these content with more detail. Okay. Any questions before we end? Would it be possible for students to make like a Google Hangout or something to share that? Oh, yes, that's a great idea. So someone shared, would it be possible for students to make a Google Hangout or something to share the worksheets and cognitive maps? Absolutely. What I'll do actually, I'll create a Google document for you and I'll make it open and you can put anything in there you'd like as a class. Could you show the slide and take, yes, absolutely. And I'll share these slides with you too. This, yeah. So once I process this recording, I will share the recordings and the PowerPoint with all of you.